All right, I'll just wait for some more people to show up. All right, I think we have a good number of people here. So what I want to do first up is field any questions about the assignment due today for the first lab, because I think we're essentially kind of working out some of the kinks in doing um, online labs, right? So I've never done this before. So stuff which I think makes perfect sense may not necessarily make perfect sense to you, unfortunately. So um, essentially, let's see, come on, it's really hard to get out of this web browser. Sorry, get rid of that, get rid of that, get rid of that, and go to here. Okay, so let's share this. So for all of these labs, because it's really hard for you to actually send me stuff physically, and basically in terms of administrating the course, having everybody send me PDFs of their lab handouts uh, would be really challenging. All of your assessments are going to be, apart from a couple of them, the linkage um, Excel spreadsheet and the little mini lab report. Those two you will be sending to me directly by email. But for all the other labs, those kind of in-class or post-lab assignments, they will be all done in Blackboard. So for every lab, and we've got the same for this lab too, I'll show you that in a little bit, you're going to have a lab handout, right? And that lab handout is essentially the one that you would be given if you were doing this lab, face to face, like normal, essentially. And the reason why I'm giving it here is because they contain a lot of really useful information for calculating stuff, for actually doing the lab itself. So I don't want to not give you those, but I do appreciate that it can be a little confusing when you see. Um, Actually, I don't want to show you that. Don't show you that either. Come on, let's open this up. No, that's printing. Don't want to do that. Don't want to look at that either. Now you're going to have a look through. Oh, you're probably not seeing this. Phew. Right, going through all the depths of my hard drive. Easy way to join the meetings. I don't think WebEx makes anything really truly easy, to be honest. Um, so there's a couple of different ways you can join. Let me just bring this up first before I forget. All right. Yeah, so there's a couple of different ways you can join. One is by clicking on that link. Um, the link should be on. Hang on, let me go back here. In the announcements section. You might have to scroll down a bit because it's here you go. So this is the link for joining the lab. There's a different link for joining the lecture, obviously. So that's one way of doing it. The other way of doing it, which I've done before, is actually let's see if I can share you. That didn't do quite the same thing that I expected it to. Can you see me moving around in my computer? Oh, okay, that's a pain. That didn't do what I expected. All right. Ooh, let's try this. Okay, can you see what I'm looking at now? This is the lab handout for, hey, yay, learn something every day. That's super cool. Okay, so the other way which you can join WebEx meetings is you can actually download the app rather than doing it through a web browser only. It kind of varies. Some ways are easier than the other. And when you have that app open, this is kind of what it looks like, right? When you're signed into your account and you have the app on your desktop. I don't know about phones. I don't have a smartphone. So anyway, here, oh, look, there's Dr. Page. You can paste let's go back to here. Where are we at? Okay, so if you see on this announcement, and it's the same deal for the uh, other one, on here, oh, you can't even see me highlight stuff. That's annoying. You can see this meeting number or access code. And so if you copy that, which I can't do for some reason because it's something funky's going on in WebEx, copy that and then you paste it into this box when it comes to joining a meeting and hit return, it will prompt you to enter the password. So you go back to here and then you just copy and paste that meeting password into the box that comes up after this. And that will also get you into the meeting as well. I actually get a reminder from WebEx says, "Hey, you've got a meeting starting soon," and I just click on that tab, that email link, just because I'm kind of lazy. Sometimes too, I actually go from my own scheduled meetings, but you don't have that open and available to you. So yeah, those two are the main ways in which you can join. There are other ways. I'd actually recommend adding it to your calendar because then you'll get a reminder and it will bring up a little reminder with this link in it so you don't have to go scouting around for it. That's another way of doing it. And in theory, you can actually call in by phone. I've never done that before, I have to say. I don't know how that would work. I actually need to try it this Wednesday. I've got a meeting while I'm on the road, which will be interesting. So yeah, those are the main ways of joining the meeting, whichever works best for you, to be honest. It's I don't have a preference one way or another. Did that help? Cool. Okay. And again, I'm kind of learning this as I go along too. This is the first time I've used WebEx in any kind of uh, depth. So if I don't know what the answer is, I'll just get in contact with support people at work, and they'll hopefully help me out, and I'll let you know the answer. Okay, so getting back to kind of the assignments and handouts, right? So this is the um, handout for last Tuesday's lab, right? And so there's a lot of really useful stuff in here that I don't want for you not to have, but also I don't want to kind of rewrite everything just for this one semester online. So that kind of, to be honest, that's the deal behind it. But I will be using some of these questions in your online assignments. So when you get a handout and it has these questions in it, don't worry about answering these questions here. This handout is nothing that you're going to send me. What you need to do is after you finish reading all of that and watching the video or being here in person, then you go back to the folder. Where are we at? Here we go. Here. And then you go to whatever that's supposed to mean, like a quiz or a test or something, I guess, and click on that. And then you can start. I can actually begin it myself. How fun is that? Right. And all of these questions should be in the same order as they are in the handout. They're actually exactly the same questions. All that will happen is that I'll give you the data or the images or whatever, either in the question or in the course content folder for the lab, right? And then you just use these data and you calculate it. When it comes to rounding, round up to 2 decimal places, right? Don't round up higher than that. Otherwise, there's a risk that your answer would not be correct. I do give a little bit of leeway just for, you know, those kinds of issues. But there's not enough to account for if you like round up from 163 to 165, let's say, right? You'll still get that one wrong. And then at the end, and this will vary from lab to lab, right? Depending on what kind of lab it is, right? There isn't any of this in today's lab, for example, but then maybe not next week, but there certainly will be the week after that. Then I need you to go back to the lab handout, right? So there's going to be a bunch of, oh, come on, back and forth essentially between the two. And then use all of the data which you have, or the values you've generated, and draw a graph, right? And so you can draw it, on here, label the axis, put in the values, put in your mean for both and your standard error of the mean, right? So you got to do everything that's being asked of you. And the standard error of the mean, you will have calculated back in, here we go, for example, here. Like when you do these questions in the online test, then you'll use that data to finish drawing the graph, right? So you've got to draw your bar graph, bar above and below, join them together with a vertical line. Those upper and lower bars are +1 SEM, so it's your mean plus the SEM value, whatever that is. And then the lower bar is the -1 SEM, that's the mean somewhere around here, minus the SEM. Right, and so that's all you have to do, and then you can draw it on a piece of paper and take a picture of it. That's fine. You can draw in Excel if you wish. It's absolutely up to you. And then all you need to do is you go save that to your computer, go back, click on Browse My Computer or phone or whatever. I'd kind of recommend doing this on a computer if you can, and then upload it and save it. And that's basically what you need to do. So again, it's because I'm the one that's kind of making these, it always seems simple to me because I'm making them. I know exactly what I want to do because I'm kind of doing it, right? But it's not always going to be obvious to you because of assumptions that I make, for example. So does anybody have any questions about, oh, okay, cool. Hang on. Yeah, I want to leave. Go away. Go. Hang on. All right, let me get the chat back. So if you want to do it in Excel, it appears that Alyssa is the person to talk to. So that's a good way of doing it. Let's go over variance because that's kind of easy, one thing to talk about. So variance is kind of similar in a way to when we were talking about chi-squared, right? So right, instead of being expected, observed minus expected squared, what you have is your actual data point minus the mean squared. Right, where are we at? Do we still have that open? We do. Make sure you're using formulas for sample, not for population. I have a feeling Alyssa knows more about how to use Excel than I do. Oh, this is the wrong one. You don't want this. Okay, so essentially what you need to do, I think there is a way of calculating variance in Excel, just as it is. And Alyssa, if you know how to do that now, I'd be more than happy to learn it from you. But I couldn't figure out exactly which was the right one to use. And so I did it by hand instead. And so, yeah, I tried that, but there was a whole bunch of different ones, which oh, var s. Yeah, that was probably the one that I didn't do. Okay, so, oh yeah, there we go. So if you put equals in and then var.s, you can also get that from the drop down up here and extra functions. Actually, let's do that again, just in case people didn't see. So can you all see me punching around on Excel? I think you can. Okay. So if you click in equals, right, that's telling them that you're going to put in a formula. And you go up here, you're not going to have var s there because it's not a very commonly used one. And go to more functions. And that's how you can search for some of the more esoteric stuff. Right? And then up here, either you can kind of scroll through there or you can type in like what you want to do. So variance. Right? And this will bring up all of the different functions that allow you to do something with variance. Oh, hang on a second. Ooh, statistic. Ah, cool. Yeah. Well, I always knew that I didn't know everything about Excel, and this is just reinforcing that. Okay, so same deal again. It's gonna automatically select a range, which almost certainly isn't what you want. So you can either click on this button here, or you can just go over here, click once, hold it down, and drag. Right? You'll see now that it comes up as B2 to B8, which is what we actually want. And then you just hit return. Man, Alyssa, if I'd known this before, it would save me so much time. Okay, everybody do that way faster. So, and then handy dandy trick. So, if you see here, I've got my female and my male set up side by side. One thing that I've learned to do is if you keep the same spacing and format for multiple different sets of data, you can use the same functions and you can just drag them across. So if you look at this box here, now it's highlighted, there's a square in the bottom. Pardon me. Double click that and drag it. to the next one. And what that will do is it will automatically calculate the same thing for the same range above it. Right, so that's a really, really powerful thing in Excel to use. Anyway, so I think that was Gabrielle. So one answer to your question, Gabrielle, is just use Excel. That's a perfectly valid answer. The other one is you can calculate it by hand, right? And the by hand calculation is one the data point minus the mean, and you square it. Let's write this down. Actually, that's a shame. We've got to get rid of all this lovely writing now. Keep it up there and impress my kids. So variance, or in Excel terms, var s, right? That's the kind of equivalent, essentially. Is this big enough, or are we going to struggle with it being tiny? I can stop sharing my screen if that helps. Okay. I'll try and bring it up. So basically, it's going to be the sum again, right? You know, so you've got to add. Well, that's going to make it really hard, Angela, if you can't see anything at all. All right, so let's. How do I stop sharing the screen? Actually, you know, I'm not sure how I can stop doing that, to be honest. Ah, there it is. It's a big orange button which says stop sharing. Okay, let's do that. There we go. So what you've got is your, let's write it up basically, rather than using symbols, because sometimes that is easier. Right, so you've got your data points. So these are like the individual heights. So one height might be 185 centimeters, let's say. And then you subtract that, from that, you subtract the mean, right? Which is whatever the mean you've calculated for that particular vector or set of data. And then because we don't want negatives, you square that, right? So you sum all those up together and divide by n minus 1. And that's really all variance is. And really what you're calculating here is how much does my every single point differ from the mean, right? Because essentially you can draw it also sometimes it's easier to draw it like this. This isn't directly applicable to what we learn, but it may help visualize it. So whenever you draw a line of good fit, you're basically trying to draw a line that has the least difference between the line and any individual point. I'm not sure this is actually going to help, but I've already started, so I'll continue. So for this particular point, right, this is the difference between the data point itself and the mean, or the line. And then you have the same for all of those. So really, get to you in a second, Gabrielle. Let's just finish this first. So variance is adding all those differences up together to give you a measure of how much the data varies around the mean, above and below it, right? Because it's very likely that the mean is going to be the same as every data point. So you're going to have some data points above, and you can also do it if that's the mean, you're going to have data points up here and down here, right? The variance is a sum of how much each of those data points varies from the mean, because the variance is going to be greater when the data points are further from the mean, right? They're more spread out. The closer they are grouped together, the smaller the variance, and so the smaller the standard deviation. Gabrielle, what was your question?
On the Excel, when you put in the variance S
Uh-huh.
and then you put in the cells from B1 to B whatever 7, is that taking into account that sum, that E?
It most certainly is. That's why Excel.
Because when I did it manually, I didn't get that number. I got a way different number.
Okay, so what is F9? Okay, so let's share crab sticks. Let's get back here. Dum, 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 dum. Okay, let's. Okay, so the way I did it kind of in a clunky way before Alyssa told me how to do it properly, um, is that was a joke, Alyssa. I'm totally grateful for learning that. I really am. Every time I use Excel, it seems I learn something new about it. So um, what I did when I did it manually is I basically typed in a data point minus mean squared formula, pardon me, and you can see the different kind of squares highlighted here, right? So you can see that this one is for the B3 and so on and so forth, right? And then essentially all I did after that was, oops, forgot about that. Let's not do that. I added all of those differences together. I summed them and then divided them by n minus 1, which is 6, because there are 7 data points in total. So what you may have done is calculated them all separately. So you may have calculated the difference squared divided by n minus 1 plus the difference squared divided by n minus 1 and then summed those together. So in this case, the order of summation is important. So you sum all of the differences together first, and then you divide that sum by the n minus 1. And so that's why the sum part is kind of up here, and it doesn't kind of span all of this. It's not just me kind of like sloppy writing. So if you were to do it ah, like that, for example, that's telling you you're going to do all of these equations separately, and then you're going to sum them together. In fact, you might even get something that looks a bit like this, right? To tell you that you're going to be doing all of these calculations separately and then add them together. That's not the case. For variance, I'm kind of taking away some stuff there. What you're doing is you're summing the differences first, and then you're dividing that by the sample number minus 1. So that might be why you got a different number. Most likely, if you did it that way, you will have got a smaller number than the actual variance. Is that what happened?
Well, okay, for the sum, what I did was add up all of the individual heights minus the means and then multiplied it by that added together. But I get it now.
Oh, okay. Yeah, so that's kind of the tricky thing which comes back a lot in like biostatistics is typically you have to do all the operations for one thing before you add them to the other. So it's the same in chi-squared too. right? So you do the observed minus expected squared divided by the expected. That's one figure. Then you add it to that whole thing for the next thing, and so on and so forth. And so that's kind of similar to variance, where you calculate the difference squared plus the difference squared plus the difference squared plus the difference squared. You add all of those together, and then you divide it by 1. You can't kind of add them together and then square it later, right? Because that's going to give you a much different figure. Or you can't treat them as, the difference squared minus n minus 1 plus difference squared minus n minus 1. So yeah, sometimes the order in which you do things can have a really big effect on what answer you get. Or you could just use Excel.
Got it. Thank you so much.
Because honestly, you're welcome. Um, I've never done any of this by hand before. It's really in many ways teaching you it this way, step by step, is really a way of teaching you the kind of underlying principles of what you're doing. And in all honesty, I've never learned that. I took a Oh yeah, yeah, it's miserable. Tell you a funny story. When I was a PhD student, I actually thought, well, my boss told me, Matt, you should do a statistics course because we're going to need it later. I was like, yeah, 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 sure, boss. So I went to it and I fell asleep probably 5 minutes in. It was just after lunch. It was a dark room. Um, and every single class period, I just went in there and then I just fell asleep. And then I woke up when everybody finished. And so I figured this wasn't a very good use of my time. I could go sleep somewhere more comfortable. And so I stopped attending that. So yeah, I don't have much of a statistics background at all, even though I do use it quite a lot. So I just use all the functions in Excel. It's just so much easier to type in, okay, what's the standard deviation? Equals stdev, select, return, done. Easy peasy. And then to be honest, I tell you what, let's show you something else funky so Alyssa doesn't get the whole burden of teaching you this. If you want to actually draw a graph, I'll show you how to do that too, right? So you copy this. Can you all see what I'm doing? Hopefully you can. Actually, let's transpose this. Stop doing that. C, no. Here we go. So let's paste and transpose. Where's transpose? She haven't done this for such a long time. There we go. So you can do lots of different pastes, which is kind of neat. Let's copy this, and we've got to paste values, right? You're going to be doing this later on in the session, right? So you don't want to paste the cells because they're calculations, and they're dependent on their position relative to the data that they're calculating. So if you just did a normal paste, you get like some funky value, right? Which is wrong. So you don't want to do that. You want to paste the values themselves. So let's go up here, select paste values. I don't know what all these other do. Oh, never used those. Click on that. And now when you click on that number, you see it's just a number. It's not a formula anymore. Okay, now we select those. And we're going to transpose those because we want them in a different format. You could just do it by hand because you've got 2 data points. But if you had like 100, you really wouldn't want to do that. So now we go up here and we do transpose. Click on that, and now you're going to have it in the format you want. Let's put mean there, and we're using SEM as well. Right, so we're going to take the SEM as well, pardon me, and, oh, bugger, we have to paste values, copy that again, and transpose it. Yes, it is. Fudge monkeys. Sorry, trying to do more than one thing at once. Let's stick it over there. Okay, copy that again, and then transpose. All right, so now we have the 2 points of interest, right, that you want to graph. Now select those two and go up here to insert graph. Let's see what kind of.
I did a scatter.
Scatter, yeah, that's probably more appropriate. Hey, that's cool, Peyton. Don't worry about it. Check out the audio, visual, whatever later, okay? Yeah, that's appropriate because this is, yeah. I mean, you could do a bar graph too. That's kind of my default go-to graph. Yeah, that's what I want. Okay. Yeah, that's kind of weird. All right, I'm going to do a bar graph just because I know how to do those a lot easier. There you go. You can do a scatter plot too. I just can't remember how to get the values down there that you're supposed to get. You can fiddle far around with these because that's a really weird distinction. So if you right click on that and format axis. Oh, yeah, that works. Let's do, you can do 0 if you want. Let's do this instead. Don't forget, you always have to have your upper and lower big enough that your error bars fit on it. Let's kind of move this out of the way. Okay, why is this in the way? Go away. Then we want a axis label. So I like how they call them vertical now. I always used to try and remember what they were. So this is going to be height in centimeters. Yeah, yeah, there's more than one way to skin a cat, to be honest, whenever you want to do this. Really, it's kind of getting the data across in a visual format that your reader can understand, right? That's really what matters. I tend to get rid of chart titles because they annoy the crap out of me. Okay, so essentially these values here are your two means, right? So if you want to add error bars in Excel, there's a really easy way you can do that. And so you can either go up here and select error bars, and I would go to more error bars options, or you can go here to this little plus thing next to the chart, and you can click on error bars there. It doesn't really matter which. Come on. Ah, you furry turd. There we go. More options. Okay, that didn't work very well. Oh, there they go. Format error bars. This is a really arse about face way of doing it, and I'm not sure if it's because I'm. Oh, here we go. Okay, so this is what error bars look like. It's basically an upper and lower bound, and you're going to need. Got to move this over here now so I can see what I'm doing. You need to select custom. This one here, custom. So hopefully you can all follow this, and hopefully WebEx is not going to completely like move stuff around and so it doesn't line up because that was really annoying. So click on specify value. Come on. I think everything's a lot slower when I'm sharing my screen. There we go. That will pop up this box and then, oh, that's interesting.
So within those braces or brackets, I always forget what they're called.
Uh-huh.
You're supposed to put in the two SEM for female, and then a comma, and then the two SEM for male.
No way. That's
Yeah,
cool.
they're both going to have positive numbers in it because the negative one automatically knows to invert it.
So,
Because I added a negative sign first and it was like, what are you trying to subtract? And so just
okay,
they're all going to be positive. So
give me just a
um,
second. I'm trying to bring up this. Oh, God, this is like communicating by snail mail. There we go. Okay, so
so you're going to delete the one in there.
normally what I just do is this.
I tried that and it only gave me positive. I don't know, maybe I did it wrong.
Let's see. Nope. So what you might have
Oh,
accidentally done is had only one of these selected up here on the top. So if you do that, and it may be because there might be some kind of funky default that applies to your version of Excel. I've come across that before. Um, so you might need to manually change it to both. And as I think someone, I can't remember who it was, mentioned in the chat box, you can obviously change the bars to have a lighter color so that you can see the upper and lower error bars. But yeah, that's typically how I do um, error bars is I just get them as long as you have the same format for the data points that you're plotting and you have your SEMs in the same kind of alignment, I guess. Um, give me a second, Angela. Um, then those SEMs should kind of line up with those different data points. Does that make sense, Alyssa?
Yeah.
Yeah, I think there are lots of different ways of doing this, and I wouldn't profess to know to say my way is the best way. It's just kind of like muscle memory, like playing tennis. I just This is how I always do it, right? So I'm always happy to learn new ways of doing things too. Okay, so Angela, your question was the plus and minus SEM. And so what you do is you go to custom. If you use any of these error amounts, I've never entirely understood how they calculate that. So I never use them. I always do it based on my own calculations. So you go to select custom, hit specify value. Oh, now it comes up instantly. And essentially what you're doing is you're giving it the plus SEM or error bar value, and then the -1 second after that. So this box here, if you click on that box there, comes highlighted in blue. Click on that, and then you click once, hold it down like a click and drag, basically, down to the bottom one and release. I wish I had like a video camera. You can actually see what I'm doing here. And then just hit return. That will put in and let's just get rid of, well, that's not going to let me. All right, let's kind of get rid of this, for example, so we can, right, causing more problems than I'm solving here.
I think you can't navigate while you have that highlighted.
All right. Okay, so I just wanted to get rid of it so that Angela could see what I'm actually doing. So when you first open this up, it's usually equals 1 in brackets or something. But anyway, so if you want to do the negative one, say you want to have both positive and negative, which is pretty normal, you would do the same thing again. You would click on the blue box, click and drag on the same SEM figures. Oh, and try not to touch anything else while you're doing that, because otherwise it stops working. Grr. Excel is very literal. And then hit return, and then hit return again. That will put you your plus and minus SEMs. So essentially all that Excel is doing is taking your mean value and either adding one SEM to it or subtracting one SEM from it. And that gives you your upper and your lower bounds for your error bars. And to be honest, just that as is, you could tie it up nicely if you want. I mean, I wouldn't use a graph looking like that in a publication just because I hate all these bars and But in terms of information, that's all the information that you need to provide. Anything more than that is just, kind of like window glossing, right? If you want to kind of conform to gender stereotypes, you could put this in pink. If you found a pink, where there is no pink there. I don't know. I really don't care. I actually like the color blue quite a lot. But you could use orange, different shades. You can do all kinds of stuff. You can do individual columns. You can play around to your heart's desire, basically. Let's go red. Here you go. Doesn't matter. It's just window dressing, really. It doesn't change the data. It just makes it look nicer. And that's really it for a graph. So, sorry, my ears, these things aren't super comfortable for a long period. Something that looks like that, either in Excel. Oh, that's okay, Angela. I mean, I used to do that too. I just got used to doing it in Excel because it's funny because if you do this a lot, doing it, the effort of learning how to do it once is worth it. If you're never going to draw a graph in Excel again, it's probably quicker to just do it by hand. So it's kind of the investment versus payoff kind of deal. And Excel is really a big issue with that because some of it takes a fair amount of learning, and it depends on how much you're going to use it, whether it's going to be worth learning or not. But certainly, oh, yeah. And it's just repetition, right? I mean, this is a page in a data file that I have, right? And so these are all actually these two figures are just recently got published in what's called a micropub, like a little publication. But I don't know if it's hundreds, but I've got shit tons of these. It's just habit. You kind of practice, do it enough times, and then you'll end up getting good at it. But certainly once you do get used to using Excel, it makes a lot of things, anything to do with data analysis, a lot easier. So I think it's worth the effort. The same as I think using a reference manager is worth the effort because the time you spend learning it is paid back, many times over in actually using it. Anyway, that's a bit of a tangent. Anybody got any other questions about the assignment for Tuesday's lab? Let's get my chat. Where's my chat button? And again, if you did it and you did it, I don't know, wrong, and you can't change it, I don't know if you can change those files after you've submitted your quiz, because I've never done one before. And you want to change it, just send me an email with that picture or even the Excel file. It doesn't really matter, anything that has the picture in it, and I'll grade that instead of the one that you submitted, right? So I don't really mind. It's not that big of a deal. But definitely put all of, oh, I actually showed you. Ha! You could have just copied these. Dang it. My Professor Fu is weak today. Anyway, yeah, let's move that down there. Put those values into the assignment on Blackboard. Gina, what's up?
Yes, I had a question because I already turned mine in. Also, I did it by hand, and mine did not have the bars. I just put because it said on there just to make a circle. So is that okay? Or
Yeah, as long as you've got the upper and lower bounds of the SEM,
yes, yes.
that's fine.
Okay,
Really,
cool.
it's getting the information across. So this doesn't have to be a bar graph. It could be a scatter plot. But really what you need is the mean and then an upper and lower bound for the SEM for each sex.
Yes.
That's it.
Okay. Thank you.
However you actually do that, it doesn't really matter as long as um, as long as I can, interpret it essentially. What you shouldn't do is put in all of the data points and the mean. That just
Didn't do
ends
that.
up confusing things.
Okay. Thank you.
Yeah. But again, if you're worried about it, just send me if you want to do it again. I really don't mind. But it's from what you've described, it sounds like you've done exactly what you should do. Any other questions? No? Cool. Okay. So again, thank you for your patience with these assignments. Again, this is the first time I've ever taught a lab online, and so it's kind of a learning process for me to figure out what makes sense to you and what works and what doesn't. So anytime something is like, what does he want me to do? That doesn't make any sense. Or whatever, just send me an email and I'll either answer them individually or I'll send an announcement out to the class as a whole, particularly if the same points come up over and over again. It just saves, everybody gets the information and it saves me time replying to the same thing to lots of people. So yeah, anytime something doesn't make sense, just give me a shout and I'm more than happy to answer it because I know that things aren't going to be as smooth as I would wish normally. Okay, so if that's it for today, so essentially what I want to do now, and this isn't going to take very long because mostly it's going to be you doing it. We've got three things, right? We've got a lab handout. That's this using micropipetters PDF. This would be the exercise that you would be doing in lab normally, right? And so essentially what it is you would have a 96 well plate, which as you would expect has 96 wells in it, and you would be adding different volumes of dye in a particular pattern to get a certain weight. And the reason why we get you to do this is that using micropipettes or micropipetters is a learned skill, right? And unfortunately, you're not going to learn much of it online, which is a little frustrating, but you can still learn like some of the theory behind it. And it's super important in almost all biology, right? I mean, you really have to be working with elephants and their excretions to not need to work in small volumes, right? Whether you're doing kind of conservation biology or molecular biology or whatever. And so you're going to be using these things a lot, even not necessarily in this lab or this course because it's all online, but certainly in micro, certainly in any of the upper division courses I teach, and certainly a lot that others do too. So the aim behind this was really to get you a lot of practice in pipetting. And we can't do that because of the format that we're doing this in. But you can still learn a little bit about how to do it properly, what the different parts of a pipette are, and so on and so forth. And so we've also got, so this is the pipetting pattern. So let's open this up. Come on. No. It's like wearing really thick gloves doing stuff when I'm sharing my screen. Okay, so essentially this is the pattern that you're would have pipetted out. And there are a whole bunch of different volumes that you would pipette. So again, it's a very repetitive kind of muscle memory type exercise. And it's a really good one too. It really helps students. Hopefully, if you don't get it this semester, you'll get it another semester in a different lab because it really does help. So essentially, hang on, let's kind of jump in ahead a little bit. And so kind of my effort, at least, to get you some of this knowledge, skills, whatever, for using micropipettes is I put together this little PowerPoint, which is a bit of terminology, like what is the different parts of a pipette. And again, most likely you know a bunch of this already. Part of it is, and this is a tricky part, which is hard to teach online, on the pipettes you use, they have 3 digits, right, which shows the volume. And how they show that varies from pipette to pipette in terms of volume. So each pipette has a very specific volume which it will measure accurately. Sometimes they can go lower, but they can rarely go much more than a few microliters higher. Like it's just the fixed volume inside them, basically. So we have pipettes that work from anything down to like half a microliter, all the way up to, I think the largest is 5 milliliters. And they look kind of goofy. We don't tend to use those very much. But anyway, you adjust the volume you aspirate or you suck up using the button on top or the thumb wheel, and you look at the volume on the side. And so the easiest way to remember this is the top digit is the largest digit of the upper range, which is kind of like a clunky way of saying if it's a P20, measures up to 20 microliters, the upper digit is the tens. So that will read either 0, 1, or 2. It's not going to read 3 because it can't measure 30 microliters. Right? The next digit will be the second digit of 20, which will be the ones. And then the third digit in red here will be like the tenths or the next decimal place, essentially. So this particular pipette is reading 9 microliters. Actually, a little wee bit more, probably 9 point, I don't know, 05 or something, right? Because it's 0, which means it's 0 tens, which is not 10 or above. 9, which is 9 singles, so it's 9 microliters, and 0, so it's no tenths, so that means 9 microliters. This one is actually adjusted badly because it shouldn't be down that low. This reads, but it's the same principles, right? So we've got a P200. The top one would be hundreds, again, the largest digit, tens, and singles. So this would be 016, which would be 16 microliters, and that's actually below its proper range. So, whatever lab researcher is doing that needs a slap on the hand. And then the same again for P1000. This is thousands, hundreds, and tens. So this is reading 1,000 microliters, or 1 milliliter. Anybody have any questions about that? Again, it's kind of awkward doing this online, but I'm going to do the best I can. This is what can get students kind of tied up in knots as well. Okay, hang on a minute. So the middle one, the rule of thumb, and this is actually interesting because I learned this from watching one of these videos. I'd already known it, but the easiest way to describe it is for the middle one, you've got 3 digits, right? You've got hundreds, tens, and singles, because the largest you can do is 200, right? Because it's a P200. Pardon me. So the top digit is going to be 0, 1, or 2, which means 0, 100, or 200. The second digit, or the one in the middle, is tens. So it will be 10, 20, 30, 40, so on. And then the smallest one is single digits, which will be 1, 2, 3, 4, 5. And so essentially you read it like each digit in order. So this will be 0, 1, 6, which will be 16 microliters because we've got 10 and we've got 6. If this was 1 at the top, then it will be 116. And so that's kind of how it's done. So however many digits the maximum volume has, that's how the digits are laid out on the display, right? So the top digit will be the largest number, which is tens for the P20, hundreds for the P200, or thousands for the P1000. And then the next digit will be the next one, kind of down in line. Okay, good question though. The next thing that students tend to mess up a lot is, and I want you to watch these videos. They're not very long. I think like 3 to 4 minutes each. And I chose them particularly because I didn't want them to be very long because this is not a super interesting subject. Um, but this plunger or push button or whatever you want to call it has 2 stops, right? So I should have brought one home. I was in the lab yesterday doing some work. Should have brought one with me. But as you can see in the videos, pointing to my PowerPoint over here, there's a soft stop, which is kind of like that. That's the volume of the chamber, right, which you adjust with this push button or the thumb wheel, right? So that's the aspiration volume, right? It's a very soft stop, right? You see I kind of click, click, click like that. Doesn't take a lot of work, right? So that's the volume that you want to aspirate. So whenever you pipette, you push down to the first stop, put your pipette in the liquid, go up and down a few times, and then suck up and kind of like release. And that will get you the very precise volume in this disposable tip. Now to get it out, what you do is you push down to that first stop again, and most likely there'll be a very small bit of liquid in the tip left, like capillary action. It will just kind of suck its way up. So then you push it all the way down to the hard stop, which takes a bit more effort, and that will eject all of the liquid. So whenever you measure out, and this is kind of hard, right? Because you're not actually doing it, and hopefully you'll remember it for the next time you use this. When you're sucking up a volume, that soft stop is the actual volume. If you press down to the hard stop, the second stop, and then aspirate, your volume will be wrong. And typically in the genetics course, you'll see that when we do the DNA purification or whatever, and like the volumes in the tubes are really crazy. And it's like, how did you get so much in there? It's almost certainly because they were pushing down to the hard stop. So just kind of have a look around at this, perhaps listen to me chuntering along here about pipettes. And these are really like workhorses of a lab. It's very rare that you'd go into a lab, any biology lab, and not see a set of these on a bench or hanging from a wall. I want you to watch these videos. They're not very long, but they're really good. They're actually made by, one's by Eppendorf, which makes pipettes and centrifuges and other things. Can't remember who did the other one. Fisher Bioscience, I think. But they're very good, and they'll give you a good overview. And then what I want you to do is go back to your course folder, content folder, go to Lab 2, and start the quiz. And so the quiz is basically going to be asking you questions about what I just said or what you will find in the videos, right? For the most part. And then at the end, you're going to have 2 questions. And essentially what I want you to do is, don't have it open. Go on. Open. Sesame. So this is essentially another way of looking at this information. It's not a bad supplement too, along with those videos. And you can see again a nice graphic showing you how these things work. What I want you to do is essentially the last part of what you do in a lab exercise. I want you to calculate the expected weight of all the dye, all of this that you add. And so this is a little bit of a tedious kind of exercise, but I had to think of something to quiz you on, right? I want you to add all of these volumes together, and that's like, essentially 5 times 17, 2 times 18, 1 times 19, and so on. A little tedious again, but it will give you something to do, I guess. And then I want you to work out, and I'm going to give you essentially a final weight that I made up. I want you to work out the discrepancy, like the difference between what you expect and what you get. And so that's really quite straightforward. All you have to remember is that 1 microliter equals 1 microgram. That's it. And so you need to convert between microliters and micrograms, and then it's one over the other, and that should give you this value here. And those are the two last questions on the quiz. And that's pretty much it. So it shouldn't take you more than ideally, 20 minutes or so to finish this up. But if you do have problems, send me an email, and I'll be on the road tomorrow and Saturday, but I'll be checking my email in the evenings. And I'll also be kind of pottering around at my aunt-in-law's place over the weekend for the next few days. So if you have any problems, just send me an email and I'll sort it out for you. And this is due Tuesday midnight. So you do have a little while to figure it out. Hang on. That's 1 microliter equals 1 microgram. And it goes up in scales. So 1 milliliter equals 1 milligram, 1 liter. Hang on, is that right? Oh, I've got to work it back. Sorry, I think I'm confusing you. Let me just check. Doesn't hurt. Thousand micrograms in 1 microliter. So it's a milligram. Dang it. Sorry, Angela. So 1 microliter equals 1 milligram. Yeah. I don't know where I got micrograms from. I should have done it the other way. That's the easiest way. The easiest thing to remember is 1 liter equals 1 kilogram, because the density of water is 1-to-1 essentially with kilos. 1 liter equals 1 kilo. So 1 liter, 1 mil equals 1 gram. 1 microliter equals 1 milligram. Yeah. Yeah, sorry about that. I think that's... Any other questions? I'll double check that I got that right in the Blackboard assignment, because that would be embarrassing if I didn't. Yeah, that's right. So the U is, yeah, this is due on Tuesday. And U equals microliter. Or micro, rather. UL. I think it's getting time for a coffee, everybody. My brain is starting to not work very well. No, Tuesday at midnight. So all of the labs are due the midnight of the following lab. I know it says in the schedule it should be by the beginning of next lab. That's kind of a holdover. And also because in Blackboard, the default is midnight. So I was like, yeah, that'll do. Plus, it also gives us time to talk about previous labs in the current lab to fix any problems that might occur. All right, does anybody have any questions about either the assignment due tonight or the one due on Tuesday? All right, cool beans. Unfortunately, I don't get the afternoon off. I've got a meeting starting in 18 minutes. So I'm going to have a quick coffee and shoot up some caffeine or something so I can get my brain back in order. Awesome. So again, remember, no lab on Tuesday, and the lab on Thursday will be the Korn and chi-squared lab. right? Where we'll be going back over the chi-squared kind of process, tests, analysis, whatever you call it. All right, everybody. I'm getting kind of flaky. So yeah, I'll see you when I get back. Again, I know that this is awkward being away right before your first exam. So if you do have any questions, please do email me, okay? I don't want you to feel like you're just going into it blind. So yeah, any questions whatsoever, I can't guarantee I'll get back to you immediately, but I will get back to you in time for it to help for your exam on Thursday. All right. Stay safe, everybody. I shall see you soon. Oh, okay. Fire away, Angela. Whew. Nick of time, huh? Just about to say, oh, let's stop. What's your question, Angela? Yes. So there is one genetics lecture on Thursday, which will be from 11 till, I don't know, whenever we finish, 12:15 at the latest, most likely. And then the exam will be open from 12:15 until midnight. So that actually gives you the luxury of being able to study what we learn in class on Thursday before starting the exam on Thursday, which you wouldn't have had if this was in person. Pardon me. Cool. All right, anybody? Anybody other questions? I'm going to stop sharing this because you don't want to see what I'm reading on the internet. All right. Take care, everybody. See you soon.